Okay, well, welcome back to the second installment of our 2012 Steenbock lecture series. It's a series of two, so we'll have to let Jack go home after this one. But yesterday, I reminded us of the legacy of Harry Steenbock, the great scientist and humanitarian after whom this lecture series is named. And I introduced you a bit to Jack, who has really made some fantastic and impactful contributions to the fields of cell signaling and bacterial pathogenesis. And as we'll see today, the hits keep on coming. And yesterday, he told us a bit about phosphatases and introduced us to some novel classes of phosphatases that his group has discovered over the years. And today, he'll take us to the other side of that reaction scheme and tell us about some brand new kinases, which his group has just uncovered, and that was published in Science just last week. So with that, please again join me in welcoming Jack Dixon.
Well, thank you. I'm delighted to be here. I've had a great time visiting with everyone at the institution the last 2 days. Just terrific. One session after the other, expanding my mind in great ways. So I really appreciate the invitation. And I think, as Dave said, Harry obviously was, he was the man. He really got this rolling, and it's great to be, to honor his legacy. I'm going to tell you a recent story from the lab, and I'm going to give you the punchline first, and then I'm going to sort of work backwards and show you some data after we do an introduction. So, in the 1980s, I started reading a lot of papers on ectokinases. Kinases are found outside the cell. And I'll show you a slide of how many papers you can pull up if you hit ectokinases. These were activities that were described in nerve cells. These were activities that were present in tissues, etc., etc. Sometimes they look like pKa, sometimes they look like pKc, sometimes they weren't characterized much beyond that. But no one really ever got down to the molecular level of where you can characterize these things from a molecular point of view. I'll even recount some examples of this today. So in the back of our mind, in the back of my mind, I knew for years that and I'll show you that casein is a phosphorylated protein. Okay, it was this report in 1883 by Hammerstein that basically demonstrated that whey, milk, what a better place for milk and dairy than right here, okay? That if he isolated the protein from milk, which turns out to be casein, it had phosphate in it. In fact, it had a fair amount of phosphate. He did 2 or 3 different methods. And it's a paper that's in German that I failed the German exam many times when I was younger, and so I had some help translating this. So we knew that casein was a secreted protein as well, and it's always been in the back of my mind. How do you account what are these ectokinases? My thought was that if you phosphorylated casein, you've got to do that somehow. Casein is on its way out of the cell. So casein was the first protein that was shown to be phosphorylated. Okay? And what I'm going to tell you about today is a small family of kinases, not your typical kinases, sort of off the beaten path, atypical protein kinases. And these are also, quotes, secreted from the cells. I use the word quotes. In fact, we know they're put in the lumen, and probably the phosphorylation occurs in the Golgi, substrates are put in the same place. And that now puts out a phosphoprotein outside the cell. We can actually detect the kinase outside the cell. So that's the take-home message. This is the first time I've given this talk. I can guarantee you there'll be more questions and answers for me because we're just at the beginning of this. So the cool part, and these are really all champions, were Hammerstein, that basically makes this observation in 1883. Then we fast forward to 1954, okay? And in 1954, Eugene Kennedy on the left there takes an extract from, I think it was liver, and again, based upon the fact that Hammerstein had shown that there was phosphate in casein, Eugene Kennedy's laboratory looks at the incorporation of phosphate from ATP as casein concentrations are increased. So this is the first example of a protein kinase activity. So Eugene Kennedy did that in 1954. Okay. Casein, as I said, is a secreted protein, and the question comes in this lovely tree that Jared Manning and Tony Hunter put together that consists of approximately more than 580 protein kinases, and each of these branches on this tree represents an individual member of the human so-called kinome domain. And if we ask the question, how many of these kinases are secreted from the cell that we know about, these are the typical, classical protein kinases, the answer is none. 0. Okay? So, it was very helpful having this tree, and basically one can easily say, well, you probably haven't studied 580 of these, so how do you know it's really not? And the answer is we really don't know that it's 0. But of all the ones that have been studied so far, all the phosphorylation we really know about takes place inside the cell, okay, in the cytoplasm. So these kinases are by and large in the cytoplasm. And you can say, well, what about receptor-like kinases? They have a transmembrane domain, an extracellular domain, but the phosphorylation, again, always occurs in the cytoplasm of the cell. So if you type in ectokinase in Google, and basically just look, you get about over 100,000 hits in 0.3 seconds. Okay? So there's your reading for the weekend. Okay? So there's a lot going on there. And as I said, these have never been molecularly characterized. Okay. Enzolpina. So people have been studying the phosphorylation of casein for a long time. In fact, those of you that follow the literature on kinases probably know that there is a protein that was known as casein kinase 1, casein kinase 2, because it worked on casein. Now that we know that it really doesn't work on casein, some years ago the names of these kinases were changed. So PETA basically has studied a kinase, Golgi casein kinase, for really 15 years. He's done the most work on this particular protein. And again, this is found in breast milk and that kind of thing. It strongly prefers manganese and cobalt over magnesium. Mo and I talked about this briefly the other day. Extremely insensitive to staurosporin, whereas most protein kinases are inhibited by this drug. And it specifically recognizes the consensus sequence serine, X, any residue, and glutamate. Okay, and you can also it will also recognize instead of glutamate a phosphoserine at this particular site. So we knew a lot about that when we started. Okay. The Golgi casein kinase is an orphan enzyme in the sense that it has really never been molecularly identified again. It's been described for many years in basically lactating mammary glands. It's an abundant kinase, and it's Tina and his colleagues showed that it was a Golgi fraction. It's secreted into milk, so you can actually go down to the store and go down to the natural food store where they have to pasteurize the milk and buy some milk, and it's got casein, Golgi casein kinase in it. I'll show you the experiment in a moment. Now, the other champion is Ken Irvine. Okay? Ken Irvine is studying a fly protein that is involved in pattern formation in flies, in polarity. And Ken Irvine basically shows in 2008 that this protein, called four-jointed, has a signal-like sequence, a long sort of domain shown here in yellow, and a domain that's simply defined as a C-terminal domain at the C-terminus. But Irvine shows in a very nice science paper that this particular protein has protein kinase activity. So this had all the right features for us, okay? Meaning to say, it had a signal sequence, it had no other transmembrane domains, and it had this domain that Irvine had characterized as being able to phosphorylate a protein, Cadherin, a protein that would fragment of it that would be found outside the cell. So basically, we took the four-jointed protein and we just carried out a simple blast reaction. Here's the corresponding four-jointed protein, and we identified a number of proteins called FAMs. That means these proteins are virtually uncharacterized. Family 198A and B, family 20A, B, C, and interestingly enough, a bacterial protein kinase that phosphorylates itself. Again, a lovely paper showing that hip A will carry out this phosphorylation. So it suggests that, in fact, this domain is a key domain in being able to transfer phosphate. But if you basically carry out the same blast reaction, you'll note that you didn't pick up any of the conventional protein kinases. So this is far enough off of the traditional tree of protein kinases that you don't pick this up. So now I'm going to focus all of my attention on FAM20C. And the reason I'm going to do that is that we express all of these proteins in mammalian cells because they're full of disulfide bonds, and they're also glycosylated. So we can make them by epitope tagging the C-terminus of these proteins with a flagged epitope tag, so we can pull this down with an antibody from media, etc. We expressed all of these, and in fact they were all expressed, some better than others. Some were secreted from the cell, some were basically stuck in membranes in the cell. These all have hydrophobic-like sequences at their N-terminus. And we began, as Hammerstein did in 1883, with casein, asking the question, will any of these proteins phosphorylate casein? And the answer is the only one that will phosphorylate casein is FAM20C. I mean, that was great news because, you've got a protein, you think it's a kinase, you don't have a substrate. So I can't tell you that any of these are kinases other than hip A, which phosphorylates itself, and FAM20C, but my bet is that these, including four-jointed, that Irvine showed, are all, in fact, kinases. So we're going to focus on FAM20C. So Benny Tagliabrachi, one of the graduate students, postdocs in the laboratory, basically, along with Sandy Wiley, localized FAM20C. Again, this is a fusion protein that we're overexpressing in cells. And this is the staining you can see. This staining you see here in green corresponds to this marker protein GM130 that basically stains the Golgi. And this is the merge between the two, suggesting that they co-localize very well. These expressions are carried out in 293 cells, but we can use a variety of cells. And we see that basically the extract contains, meaning to say the extract of the cell, things that are in the cell, contains some FAM20C, but a lot of it is found in the medium. Now remember, this is overexpressed, so we don't really know that in the natural situation, if this material would normally be secreted from the cell, or we're just overloading the secretion machinery and some of it's coming out. Okay, so this is the purified protein. It's actually pretty good. Purified, as I told you, with the epitope tag. So it works on casein. So the next question we really tried to ask was, what substrates will this particular protein kinase phosphorylate in addition to casein? And one can buy these peptide arrays. I think this is about 800 peptides or so. They're all done in duplicate. So two spots right next to each other basically means a positive hit. If you're missing the second spot, it means it didn't repeat. And you can see that there are a fair number of small peptides. These are a bunch of known phosphorylation sites in phosphoproteins that are found in the cytoplasm. But you can see that there are several of these that are clear-cut hits in our peptide array. So then we were happy about that. When we interrogated those peptides that in fact were hits, what we could see was that there was a real preference for serine phosphorylation on those peptides. Several residues and acidic residues work pretty well, but again, a very specific requirement for SXE. Okay? Now you may remember that's the same motif that Pina observed in the Golgi casein kinase from milk. Okay? So it turns out this is a rather specific recognition by this kinase. We basically produce this peptide, which corresponds to one of the phosphorylation sites in casein kinase. You can see that the serine, the SEE sequence is phosphorylated, effectively again with ATP. And if we change the E to D, pretty conservative change, not much phosphorylation. We change the S to threonine, no phosphorylation. Threonine to threonine and aspartic acid doesn't work, and tyrosine doesn't work. So it really has major specificity for this SEXE sequence. We're pretty happy about that, okay? Is the FAM20C the real casein kinase? And the answer is, that's exactly the answer, okay? Yes, it is, okay? I'm going to show you some evidence for this. So the first thing Benny basically did was to show that if you started out with casein, purchase this, add the kinase, purified kinase in vitro, and basically we could see as the phosphorylation proceeded, shown here on the bottom part of the slide, we see these shifts in the migration of the protein as more and more phosphates are accumulated in the casein. We took a guess at a highly conserved residue in the kinase that was invariant. We actually mutated that. We know this kills kinase activity, as you can see here, and it kills phosphorylation as well. This also suggests that the kinase that we're adding, purifying from the mammalian cells, is what we think it is rather than some extraneous thing, because when we put in the dead kinase, basically, phosphorylation doesn't happen. So it works on casein in vitro. Now, what if you co-express FAM20C along with the kinase in the mammalian cells, code express the two together. And the answer is you get a shift in this band. And if you treat this band with phosphatase, you bring it back down. So this shift in the band here that we see taking place inside the cells is associated with phosphorylation. So what we think is going on there is as follows. And I'm going to spend a minute or two on this slide. So we think that basically casein is being synthesized, and because of its signal sequence, it's being thread into the lumen of the ER, where it will ultimately probably reside to some degree in the Golgi. And likewise, we think that FAM20C is, again, with the hydrophobic signal sequence at the N-terminus, will thread into the lumen as well. And so this is sort of depicted as the lumen, the Golgi compartment, etc., etc. We really don't know the exact place that all these phosphorylations take place. And then in the presence of ATP, and obviously ATP probably has to be pumped into this organelle as well, carries out this phosphorylation. And then we see the phosphorylated casein outside the cell, and we see some of the FAM20C kinase outside the cell as well. All right. Can you get the phosphorylation if you take the signal sequence off of FAM28? Not going to show you the data for that, and the answer is no. So the signal is essential for basically partitioning. Do you get the phosphorylation in vitro when you basically make a catalytically inactive FAM20C? And again, the answer is no. So this is basically what we think is taking place in vivo and producing the phosphorylated casein. And this sort of fits all the bills. Basically, these are going to be extracellular kinases of some sort, and these are going to be extracellular proteins that one finds in milk, like casein. All right, a couple of other points. So you remember that Pina and his colleagues demonstrated that this kinase preferred manganese and cobalt over magnesium. And in fact, our recombinant protein prefers manganese and cobalt over magnesium. And likewise, you may remember that staurosporin, which is a very effective inhibitor at this concentration, this high concentration for all the traditional protein kinases, is not much of an inhibitor for the Golgi casein kinase. So again, this has all the properties for the Golgi casein kinase that Pina worked on so hard. Okay. All right. So in the year 2000, Burnett basically published a paper in the Biochemical Journal where he fractionated the Golgi casein kinase from milk 80,000-fold, and he isolated a Kinase stain band, and that Kinase stain band, all the peptides corresponded to alipoprotein E, and he suggested in the article that the Golgi casein kinase is still a minor component from his mono Q column. But he attempted to purify it, just didn't get it pure. So then he took two of the steps out of the purification scheme that he had used. And here basically we're looking at activity, kinase activity in red, and basically absorption on the right-hand scale. And this is over a Cephalose column, which was used in the fractionation. And you can see that the kinase activity basically peaks, as you can see in red, and some fractionation taking place as the protein. And by this time we had an antibody against FAM20C, and this is a blot of FAM20C, and you can see that the fractions in fact correspond, the immunoreactivity corresponds exactly with the fractions that have the casein kinase activity. And we took those fractions and basically purified them again on a mono Q column, as had been done before. And without going through the details again, the three fractions that contain the immunoreactivity also contain the kinase activity. So we were pretty sure now that this was really the Golgi casein kinase. Okay. Now this is another amazing story. So this was a report in 1989 by a physician known as Raine, describing a phenomenon in children known as Raine Syndrome. And even though one didn't know the function of this protein, by sequencing, by basically genetics and sequencing data that was done later on, one could demonstrate that there were mutations in the FAM20C gene that led to these phenotypes in children. In fact, most of the children die before birth or shortly after birth, and some of the ones shown here are in fact kids that have survived a few years into their life. Okay? So these mutations that were reported these mutations were found in FAM20C and results in this autosomal recessive osteosclerotic bone dysplasia. And these children have characterized by abnormal hardening and an increase in the density of the bone. In fact, their skulls sort of overgrow, and this is hard to see in any of these things. They sort of overgrow in terms of the bone density. Death usually takes place in the first few weeks of life. Calcification in the brain, in this area of the brain, is commonly seen in these individuals. So this is a mutation in the kinase. Okay? So James Ingle decided to make each of these mutations that were seen in the children, and in fact, two of these mutations are found in the children that are alive, and these others were in fact identified in the children that died shortly after birth. That kind of thing. And in every case, again, I won't belabor this, but in every case, what we see is that we can synthesize the proteins in the mutants, okay, but they basically cannot traverse, they don't get secreted well. These are controls, a wild type control, and inactive protein, inactive kinase you can see here. And you can see only two of these basically make it through the secretory pathway very well. And we used another bone protein called osteopoietin that I'll come back to in a moment, but we could have used casein here as well. And you can see that the only two proteins that are partially phosphorylated are these two at the end, and in fact, these correspond to the mutations that are found in the children that survive the longest. Okay? So this makes it look like that the protein kinase activity and the ability of these proteins to traverse the secretory pathway correlates with the severity of this particular disease. And you can see that many of the mutations are in fact in the kinase domain. So we like that idea. Okay? So if we line up that, remember this is a little family of proteins, if we line up the FAM20A, B, and C, we can see they all have the same basic geometry. They all have hydrophobic signal sequences, parts of the amino acid sequence that don't show much identity, and then a high degree of identity at the C-terminus. Okay? We tried actually making just the C-terminal fragment, but with no success so far in E. coli, that kind of thing, because we'd love to do a crystal structure of this. Now, the next few slides were really carried out with Nick Grishin and his look back, and we can ask, can we find some of those key residues in the kinase, in traditional kinases? And the answer is we can. In fact, the catalytic aspartic acid that's found in traditional kinases appears to fit here. Okay? The metal binding residues are shown here, okay? Etc., etc. Now, I'm not trying to make an important point here. I'm simply trying to say that we can identify residues that we think correspond to key residues found in some of the classic kinases. But the residues that surround these are so poorly conserved that basically this is subject to interpretation. Okay, but we think that at least it points to the notion that the key residues in the kinase, in fact, are conserved. So this is Nick and Lisa's work, and basically they're collaborators on this effort, modeling the FAM20C over the HIP-A sequence. And again, this is probably a very poor model, but if we focus our energies on the active site, we can see that there are a number of residues that basically would be predicted to be involved in binding the metals involved in this particular case. Another basic charge here. And so what Nick and Lisa did was to compare this with the sequences that surround the active site in protein kinase A, the protein kinase that Susan saw initially 20 years ago. And then we can sort of compare how these two sort of look. And I think the residues surrounding the metals are somewhat the same. Basically, the basic residue here and the corresponding basic residue here probably playing a similar role in the binding properties as well. So we think that basically this provides a rough indication that the likely fold for this particular protein kinase is going to be similar to the traditional protein kinases as opposed to something completely in left field. But again, we really need an proper X-ray structure to deal with that. Okay, so we started looking at proteomic data. This is not something we did, and this is just stuff in the literature. And the things that were striking to us was that we looked at phosphoproteomic data. This was done in 1910, reported in plasma, and phosphoproteomic data that was found in cerebral spinal fluid. And we simply looked at the number of reported examples of peptides that basically had the serine X glutamate sequence in either of these two tissues. And in fact, about 75% of the secreted phosphoproteome represents phosphorylations of these residues. Okay? So this suggested to us that there were likely other substrates for FAM20C. And again, this has been known for some time, since 2003. And it's known that, I think this is on chromosome 4, that there is a cluster of genes that probably all arose from an ancestral gene that include such proteins as casein. There are several casein genes in this group. Osteopoietin, a bone-specific protein, is in this group. There are a bunch of proteins that are involved, siblings that are known to be involved in bone and teeth formation. And in many of these cases, these proteins, as I said, have known phosphorylation sites. In fact, some of those phosphorylation sites are on the SXE motifs, and we think that this is playing an important role in basically binding calcium. So the phosphorylated peptide probably binds calcium, and that calcium is important in milk, and that calcium is also probably important in bone and teeth formation. Okay? So basically, we're going to take 2 or 3 of these proteins, and I'm not going to belabor this, and show you that these can be substrates for FAM20C. I think we're going to look at osteopoietin, which is the top one, dental matrix protein, one we talked about to some degree. There are other extracellular proteins that fall into this category. So these are some examples of the phosphorylation. This is osteopoietin, shifting in the band to higher and higher molecular weights. It's again an in vitro experiment. This is the phosphorylation that we basically see. One of the other matrix proteins shown here, again we see increasing phosphorylation. This doesn't band shift nearly as well. Again, the dead mutant doesn't work. And we can go on and on with this. Okay? these are the DMPs, and again. And what you can actually see is that as the phosphorylation gets to be real significant in terms of amounts of phosphorylation, the Kinase dye doesn't stain so well the proteins anymore. So it's really phosphorylated to a high degree, I think. And so the FAM20C also phosphorylates these sibling proteins, and again, I won't go through this example, but we can again show that this shift in the gel is phosphate specific. And again, another example over here with the shift in the gel being phosphate specific. And the interesting thing, if we just look at some of these proteins such as osteopoietin, they all have signal sequences. They're going to be outside the cell. They're going to make up the composition, the organic composition of bone, okay, and teeth. Some of them have RGD sequences that will interact with cell surface integrin-like receptors. And they're just full of these serine X, serine XE, etc., etc., etc. And this is true of the entire family. Okay. So this is really about where we are. FAM20C we think is a secreted protein kinase. We guess that the whole family of these are secreted protein kinases. FAM20C is the real Golgi casein kinase responsible for phosphorylating casein, the first protein ever to be shown to have phosphate incorporated in it. It's probably the true casein kinase that Pina and his colleagues have worked on for a long time. In addition, these are all substrates, as I said. There are numerous examples of the substrates that basically have these SXE motifs that are mutated, and mutations in FAM20C looks like it. They inhibit secretion and kinase activity, and those result in the disease. So what we think is going on with the bone mineralization thing is we think there's a competition for calcium. And in fact, this has got to be regulated in a very specific way so that these bone proteins are going to make up sort of the organic part of bone, if you would, are a mixture of the phosphates that are attached to these XXE motifs. We haven't really shown this rigorously, but others have shown that these motifs, in fact, bind calcium. And we think that this probably competes with hydroxyapatite formation, so that if you do not chelate all the calcium, it ends up as calcium phosphate. And this is that brittle part of bone that basically one is described in the Raine Syndrome patients. And really, it's a proper sort of mix of the organic and the inorganic part that results in. Now, I actually don't know anything about bone at all. Okay, so if you tell me this is wrong, I would be happy to chat with you about it. So these are the people in the lab that contributed. Vinny's really did most of the work. James did the mutations in the Raine Syndrome. Sandy helped with the microscopy. I didn't talk about Wen's work, but it turns out that one other member of this family, in fact, isn't a protein kinase. It turns out to be a kinase that phosphorylates a sugar residue on a glycoprotein. Okay? So Wen's working on that. Carolyn sort of helps oversee the lab. Jun Yu helped with all the structural stuff. And Lisa and Nick Grishin were terrific collaborators on the bioinformatics as part of this. So again, I want to thank you guys for being such great hosts, and be happy to answer questions. Questions? Yes?
Two quick questions. One, I think you showed these in fruit flies, but fruit flies don't have bone and milk. So do we have any idea what the FAM20s are doing outside of mammals? The other question is, what's the point of phosphorylating casein? I mean, if it's already in milk, what's that for?
Okay, so the two questions. One is what's this doing in flies? Turns out this gene in flies is one of the classic examples of a developmental gene. Four-jointed actually means that the leg of the fly has 4 joints instead of 5. So this is a so-called polarity gene in flies. Our guess would be, as I mentioned, Irvine showed that it would phosphorylate fragment of cadherin. We really probably don't know all the substrates that it will phosphorylate, but probably in phosphorylates, those substrates, again, we predict at the outside of the cell, and that somehow, I don't know how, sets up this polarity so that we get 4 joints instead of 5. Okay? Turns out there's one example of this in worms as well. Worms don't have bones either. We've actually produced that protein and asked, does it work on a carbohydrate or does it work on a protein? It works on protein. And we think we can spot a substrate that looks like sort of a worm version of one of the, quotes, bone proteins, and just don't know any more about it than this. But we think probably it does serve some function. In other words, they don't all have to work on bone. And your second question was?
Oh, what's the point of phosphorylating?
Casein, yeah. Well, I think, again, don't know that answer, but I think the phosphorylation of casein lets you bind calcium in large amounts. And I think nutritionally, having calcium and phosphate and a ton of protein in milk with some lipid in there is probably why we're all here.
You can't form the micelle without the phosphate.
Right, can't form the micelle without the phosphate. And that probably has to do with chelating the calcium, would be my guess. Okay. No?
Why does it prefer manganese over manganese?
That's the question I asked you, Mo. Mo's answer, I'll tell you Mo's answer. We're going to go back and look for this. So, Mo suggested, I don't know the answer to that, okay, but he suggested that the residues surrounding the active site may be such, suggested even that it could be a sulfhydryl reagent, I think, that could have a preference for the manganese. I don't know enough about this.
Well, in that model, is there a cysteine nearby?
I'll have to go back and look. There isn't in that model, okay? But again, we tried to pick out just a few residues that we had some positive feeling about what they were doing.
So all your assays are done with manganese, I think.
Yes. Except a couple we did with cobalt, but we routinely just do it with manganese. Yes?
We did some earlier work on casein manganese, and we realized that cobalt Dixon actually activates the enzymatic significantly. And he would actually refer to that as cobalt activated kinase. And it turned out that actually when you bind the casein or phosphate, change the conformation of the protein, it makes a better substrate. So to lower the Km value for the casein or phosphate. And we did dephosphorylate phosphate, and we showed that actually that's the case. That cobalt
Great. Yeah. I think probably in this case too, cobalt probably binds to could bind to the substrate, but it probably also binds to the enzyme. Yeah, so thanks. I didn't know that. Yes?
Jack, you mentioned HIP-A at the beginning.
Yeah.
Which is a really interesting protein.
Yeah.
You mentioned it self-phosphorylates.
Yes.
So, do you know what the regulator?
And there's a structure on that, by the way. That's over here is the model. Yeah.
Do you know what the regulator is of that phosphorylation? Is it always on?
I don't think that's known. Okay. I think the reason they did the experiments with phosphorylated itself is you could add ATP and you didn't need to know the substrate. I'm not sure they know the substrates. It's one of these toxin, antitoxin kind of things. So the logical thought would be it would be the antitoxin that might be change the binding and that kind of thing, but I don't know. Yes?
Does there exist, or do you think there exists a secret phosphates?
That's a good question. Okay, we thought about that. Okay. I think Judith Kimball basically asked a version of that question yesterday, and I would have told you maybe a year ago that no chance of that being the case, but I wouldn't exclude it. Okay, maybe you have to. And what's interesting is in the carbohydrate phosphorylation. We see the carbohydrate phosphorylation taking place and being very abundant early in the maturation process of the carbohydrate, and then it disappears. And you say it's much less. So there would be a perfect place for the phosphatase to take that off. So maybe once you do your job, you basically want to get rid of it. So we've been going, full blast on this, so we really haven't got to that question, but that's a good question, real good question. Yes?
Jack, I saw one slide that showed the Western, the band is higher, lower.
Yeah.
And we meant this problem. How to explain that?
We think two things. First of all, we think all of the ones that we've looked at and expressed are glycosylated proteins. And it turns out that in a large number of cases, not always, phosphorylation will actually cause a conformational change or something in the protein and cause the protein to run larger, apparently larger on SDS gels. So those shifts in the gel we think are due to phosphorylation. And the reason we think that is if we take the shifted band and treat it with phosphatase, we can basically move that phosphorylated protein down on the gel back to the native spot. So we think the shifting is actually the phosphate being on the protein to move to a slow mobility, treating with the phosphatase. Basically, it shifts back down to the normal mobility.
What group is being phosphorylated? Serine, threonine, tyrosine?
Serine.
Serine.
Yeah, and as I showed you in that little peptide, it doesn't like threonine, it doesn't like aspartate for glutamate, so it likes that serine, and the serine becomes serine phosphate. And our guess, Mo, is that serine phosphate and the glutamate form a very good binding site for that calcium. We haven't really got there yet, but I bet you that's what's happening.
Actually, that distinguishes from the other Casein kinases, where we can tolerate aspartate and also we can take really
So it'll be different, yeah. Yeah. Yes, back in the back.
Does the recognition sequence need to be in a disordered part of the protein, or can it be in a structured part?
Well, most of these proteins are not real well structured. In fact, we have many of these phosphorylation sites. So my guess is that unfolded is probably better. Now, will you have some cases where those residues are in a folded protein and are right on the surface? It probably will. But I think the vast majority of these proteins are going to be low or no, not a lot of structure. Yes?
The Raine Syndrome, is that dominant?
Raine Syndrome, no, it's recessive. Okay, so you need two bad copies. Yeah. Yes?
So I'm curious as to whether you've ever tested any of the bisphosphonate drugs to see whether it either inhibits or activates or has any influence on phosphorylation of some of your substrate proteins that are associated with bone metabolism.
We haven't tested those. We read a lot about them, but we haven't tested them. It's a good question. Okay, thank you all.